Okay, hello everyone. I'm Siti Sarah Hanum together with my members Nurul Ashikin and also Pirsana. Today we would like to review on an article entitled A New SNP Genotyping Technology Target SNP Sequence and its Application in Genetic Analysis of Cucumber Varieties. Okay, these are the contents that we will deliver throughout this review. We start off with introduction. Okay, variation at a single base pair is called a SNP or single nucleotide polymorphism. SNPs are ideal markers for genetic background analysis due to their advantages in high throughput detection and also easy to integrate genotyping data. There are several traditional SNP genotyping methods based on electrophoresis systems such as CAPS, DCAPS, and ASPCR. Okay, they are commonly used in genetic research. The invention of certain high-throughput SNP genotyping techniques, such as the Gene Chip Microarray or the CAPS platform, has been supported by ongoing developments in high-throughput sequencing. Chip-based platforms require enormous amounts of time for SNP assay designing and are only suitable for genotyping hundreds of samples with thousands of SNPs. CAPS systems also need relatively high cost for genotyping each sample. These SNP genotyping platforms often find that some SNP loci cannot be well detected and genotyped because the flanking regions of these SNPs are not conserved. This causes many false positive or false negative results in SNP genotyping. Therefore, it is necessary to use a new technology that is cost-effective and highly accurate for genotyping genome-wide SNPs. Next generation sequencing, NGS, approach is currently broadly used. There are few researchers that have devoted to the study of comprehensive genomes variability and the creation of ideal SNP loci that harbor preserved snipping errors with special genome PCR amplification. A new method called target SNP sequence, which combines the advantages of high throughput sequencing and multiplex PCR amplification, has been developed by using genome-wide perfect SNPs, which meet the requirements of harboring conserved flanking sequence and being captured uniquely in PCR amplification. Okay, so this approach could also gain hundreds of SNP genotypes by sequencing the SNP region with an average of 1,000 times coverage in a short time and at a low cost. Okay, in China, it is the world top producer and consumer of cucumber, cultivating more than 1.24 million hectares. But the rising amount of commercial cucumber varieties every year poses a great challenge to the monitoring and conservation of plant diversities in China. In spite of some cucumber DNA fingerprinting studies that has been recorded through simple satellite repeat technique, it was still hard to obtain high throughput genotypes. So in this study, they applied the target SNP sequence genotyping technology with 163 perfect SNPs to establish the DNA fingerprint of 261 cucumber varieties and also to reveal the population structure and genetic features in cucumber varieties. Now we move on to methodology on how they conducted this study. I will pass to Prasanna to further explain about it.
Okay, I would like to focus on the methodology part of this research where next, there are 5 major methods that have been conducted in this experiment. The first one is the discovery of genome-wide perfect SNPs in cucumber. Second, construction of target SNP sequence library. The third, the target SNP genotype calling. The fourth is plant materials and DNA extraction. And the last one is the data analysis. Next, so the first one is the discovery of genome-wide perfect SNPs in cucumber. So here there are like about 6 criteria of SNP sites for perfect SNPs have been stated. The first one is only the biallelic SNPs should be remained. The second, 30 base pair flanking sequence of a SNP are used to map to a unique region in cucumber associations, and there should be no indels, means the insertions and deletions, and SSR or the other SNP locus. The fourth one is the heterozygosity is no more than 0.2, and the missing rate is less than 0.2. The MAF should be above the 0.4. Then, finally, the all perfect SNP loci will be sent to the mold breeding biotechnology company in order to conduct the multiplex PCR primer panel design. Then, okay, you can see here the DNA give test to what's that to resembles the what I gonna explain later. So here, the second method is the constructions of the target SNP sequence library. In these methods, there are 2 rounds of PCR have been conducted. Why 2 rounds? Let's see the purpose of these 2 rounds of PCR. First round, to capture the target SNP locus and also amplify 200 till 800 base pair sequence in DNA samples through multiplex PCR. Then the second round is to distinguish or differentiate each DNA sample by the addition of the unique barcode adapter. Okay, next, let's focus on the materials and methods for the first round of PCR. So the materials that have been used in this experiment for the first round are the total, they use a total volume of 330 microliter containing 50 nanogram DNA, 8 microliter SNP primer mix that consists of 0.2 micromole per liter and 10 microliter 3M enzymes, which is the Genoplex that is obtained from the mold breeding biotechnology company. So what means of 3M enzyme is actually a mixture of 3 kinds of enzyme, that is including 2 mutant tag enzyme and 1 high fidelity thermostable DNA polymerase. Then they use 2 units of enzymes per reaction. So let's see the methods. As we all know that PCR is known as thermal cycling, and there are 3 major steps, which are the denaturation, annealing, and the final extension. Here they run at 95 degrees Celsius for 5 minutes, which is for the denaturation part, and followed by 17 cycles at 95 degrees Celsius for 30 seconds and 60 degrees Celsius for 4 minutes, which is for the annealing process, and the final extension at 72 degrees Celsius for 4 minutes have been conducted. Then the magnetic bead suspension has been used for the collection of PCR products, and 80% of ethanol has been used for the purifications of the PCR products. Okay, next move to the second round of PCR, where the materials used are next 30 microliter PCR mixture that consists of 11 microliter purified PCR product that is obtained from the first round of PCR, then 10 microliter tag enzymes, 18 microliter pure water, and 1 microliter barcode adapters that composed of forward and reverse primers. Then the methods is a similar process, but there is a changes in the temperature and the time, where they run at 95 degrees Celsius for 3 minutes, that followed by 7 cycles of 95 degrees Celsius for 15 seconds, 58 degrees Celsius for 15 seconds, 72 degrees Celsius for 30 seconds, and the final extension conducted at 70 degrees Celsius for 4 minutes. And the magnetic beads have been used for the enrichment, and 100 microliters of 80% ethanol used, and 23 microliter of 3 hydrochloric acid buffer, which is at concentrations of 10 millimolar at the pH 8.0 till 8.5 in order to wash 3 times. Then, consequently, the DNA library was sequenced on an Illumina high sequence X10 platform in mold breeding biotechnology company. And then they also sequenced the DNA of Chinese cucumber in breed line 9930 as a positive control in order to verify the accuracy of the target SNP sequence. Okay, next move to the third method, which is the target SNP genotype calling. Here we can see there's a figure that's adapted from this experiment, which is shown the procedures of the target SNP sequence in the genotyping 261 varieties with the 163 perfect SNP. So here we can see there is a process where it's a genomic DNA, and then they obtain the primer piece, they prepare the primer mix, and then they conduct the first round PCR to capture the target SNP locus through multiplex PCR. Then the PCR products enrichments have been conducted and continue with the second round PCR is to add a unique barcode adapter for each DNA samples. Then they conducted the library. They construct the library. The constructed library was sequenced on the high sequence X10 platform. Then finally the previous slides. In this case, the specific SNP variant bases for each variety were identified by analyzing the in-house barcodes against the raw sequence reads. This is conducted to ensure the accuracy of SNP genotypes. Therefore, the specific SNP variant basis for each variety has been identified by analyzing the, as I mentioned just now, the barcode assigned raw sequence reads. Next. So the fourth method is the plant materials and the DNA extractions. So the purpose is to evaluate the novel technique of the target SNP sequence. Therefore, a total of 261 cucumber varieties have been selected in variety identifications. So the process involved here is newly expanded young leaves grown from 30 seeds have been used, which is to increase the genotype accuracy of each cucumber variety for DNA extractions. Then a plant DNA magnetic bead kit have been used for what? To store the total genomic DNA that has been isolated from the freeze-dried tissue in 96 well plates. And lastly, a qubit 2.0 fluorometer with a Quant IT double-stranded DNA HS assay kit have been used for the concentrations of the all extracted DNA before the targeted SNP sequencing. Then the last method would be the data analysis. So data analysis, there are 4 divisions have been focused here. So the first one is genetic diversity analysis of target SNPs. The second is a population structure analysis of 261 cucumber varieties. So in the first division, the analysis used are the MEF, just now I mentioned, which means the MEF is the minor allele frequency. So these are the various genetic diversity parameters have been focused based on the MEF. The HR is the observed heterozygosity. The PIC means the polymorphic information content, and then the D means the Simpson's index of diversity. F means inbreeding coefficients. So all these calculations they performed in the Excel 2016 by using the formula mentioned in these articles. So the second divisions, the analysis or software that have been used is actually quite a lot. Let me explain one by one here. So the model-based program structure version 2.3 is used to analyze the genetic structure of 261 cucumber varieties. And then they also perform the 3 parallel runs to determine the population's numbers. That is, what's that denoted as symbol K. Then in order to verify the optimal numbers of the clusters, the principal component analysis has been performed in the respective R package. Then they use the neighbor joining tree from the MEGAR 7 which is to analyze the genetic relationship among the 261 varieties based on the genetic distance in POP-R package. Okay, then they also conduct the analysis of a molecular variants, which is new for us, because we are usually you learn on the ANOVA, right? So this AMOVA, actually they used to measure the genetic differentiations between pairs of subpopulations, and they computed in the pairwise what's that called? I don't know, FEAST and the POP-R package. Usually we conduct on SPSS, right? So it's something new. Okay, next, the third and the fourth divisions. Okay, let's see the third divisions. Selecting core SNP loci for the cucumber variety identification. So here, they use two software analysis, which is a script in Perl. The script in Perl actually they use to screen out the minimal number of SNPs for differentiating the maximal number of cucumber varieties. Then they conducted on the minimal marker. Minimal marker actually what's that? It's called. Actually, I don't know what to say. And then they continue these experiments by performing on the minimal markers. And then the second one is the selecting core cucumber varieties in the Chinese market. And here, the analysis I used are the pairwise comparison matrix. This pairwise comparison matrix actually by calculating the number of the differential SNP genotypes between each pair of varieties, and also the missing genotypes was treated as null. Then the second is the correlation analysis among the three pairwise comparison matrix by that is calculated by using this software, which is the SAS 8.1 PROC-Core. This this software is actually conducted to see the genetic similarity. So these are the overall analysis that have been used in these experiments. And next, I will pass to Nurul Ashikin to further explain on results and discussions. You may proceed, Shikin.
Thank you, Pira. So for results, as you can see, the figure illustrates genetic characterization of 163 SNPs from 261 variations of cucumber. The observed heterozygosity, as what have been presented by Pirasana, of 261 variations range from 0 to 0.64, while this value in 154 SNPs varied from 0 to 0.44 with an average of 0.17. So the PIC value ranged from 0.101 to 0.5 with an average of 0.388, while the mean value of MEF was 0.291 and varied from 0.054 to 0.496. Interestingly, the inbreeding coefficient in 3 SNP loci was equal to 1. Which indicate that these SNPs had no heterozygous genotype in all varieties. So the average genetic diversity coefficient for 261 varieties was 0.63, which indicate that 261 cucumber varieties had low diversity and high similarities in genetic background. Next. So this bar chart illustrates the distribution of sequencing, coverage and read ratios for SNP sequence target, where A indicate the sequence depth of all reads, B indicates sequence depth of 154 SNP loci, and C the 261 cucumber varieties, and D is the allelic reads ratio of homozygous and heterozygous genotype. Next. I will continue for discussion part. So there will be 4 subparts and I will further explain for each part. Okay, next. Okay, a few techniques to SNP genotyping have been described, and the provision for SNP engineering needs accelerated over genetic research. However, high cost of production and poor productivity are still problems for the current SNP genotyping platforms. Certain NGS-based genotyping approaches, as what have been presented by Sarah, have such as AMP sequence, GBTS and sequence SMP are not focusing on developing the perfect SNP loci by using variums of big data. So besides that, the probe hybridization is also needed in the amplification of SNP's primers. Therefore, in this study, a newly reported method in SNP genotyping target, which is SNP sequence, is utilized. So SNP sequence possesses a few advantages such as high accuracy in SNP genotyping, where the average reading depth of the chosen ideal SNPs is indeed over 1,000 times in this analysis. And the statistical precision of SNP genotyping in target SNP sequence was as high as 98.7%. Besides that, it possesses good repeatability, which alleviates the rate of false positive and also false negative. And it is cost effective as it only involves small costs for each sample of DNA. Next. Thus, relative to the conventional SNP genotyping approaches, the SNP sequence approach had apparent benefits in genotyping hundreds of SNPs. Next. Okay, in this study, a total of 857 gigabyte data from 182 extremely diverse cucumber accessions covering all cucumber ecotypes in China were evaluated. 3,000 cucumbers germplasms and a total of 115 accessions have been classified according to morphological and geographical properties, and 261 dominant cucumber varieties are analyzed using target SNP sequence with 163 genome-wide perfect SNPs. Next. The four subgroups of Chinese cucumber varieties are Indian group, Eurasian group, East Asian group, and Zhishuang-Bana group. So from the findings, it is demonstrated that the first two subgroups of Chinese cucumber varieties derive from the previously reported East Asian group. It is also demonstrated that the Zhishuang-Bana cucumber type possess higher inbreeding coefficient and genetic distance diverge further than the other three types. So this was in accordance with the semi-wild cucumber type reported by the previous study, where the genetic diversity of South China type and Europe type are higher compared to North China type and Zhishuang-Bana type, in which this indicates that those two types have a narrow genetic diversity have a narrow genetic background. So the close resemblance and narrow genetic diversity of cucumber variations implies that the possibility of genetic erosion in the breeding process is triggered by the narrowing of the exploitation on the new genes. Next. DNA fingerprint is conducted based on the target perfect SNPs in cucumber. So DUS stands for distinctness, uniformity, and stability. Each new candidate variety should undergo DUS testing that is known because it protects the intellectual property of the crop breeders. Because of high throughput genotyping, the SNP technology has been applied to a variety identification and DNA fingerprinting, at which the microarrays and other systems-based studies were conducted. However, wide use of this technology is difficult in variety identification and management due to the high cost for each variety, highly time-consumed, and high number of false positive or false negative caused by SNPs with various flanking sequences or specific capturing. Next. So in this recent study, a set of 163 perfect SNP loci was used, which had unique amplification and conserved flanking sequences in the cucumber genome. Then 182 cucumber accessions resequencing data sets were analyzed, and an appropriate DNA fingerprint was established for each cucumber variety by using the target SNP sequence. So 99% of 261 cucumber varieties were differentiated in order to calculate a core set of 24 SNP loci that is representing a discriminating capacity as high as 98% of the total 163 SNPs. Therefore, the obtained outcomes are essential to identify the authenticity and the measurement of the purity of various cucumbers, and also to protect the plant varieties in the upcoming generation. Next. So the prospects of SNP sequence were applied in the plant breeding system. So in many plants, marker-assisted selection (MAS) breeding has increased breeding performance at a tremendous rate over many years. Hundreds of crops' genomes have been gathered and genome-wide variants have been researched with a significant progress of genome sequencing and resequencing, causing it simple to build contact selection in the breeding via the MAS method. So in this research, target SNP sequence was shown to be a versatile, reliable, and inexpensive SNP genotyping method that could be widely used in the modern breeding system, including the pyramid breeding, background selection, design breeding, foreground selection, and a lot of validity, detection, and purity examination. So as a result, in the coming years, target SNP sequence innovation will certainly provide comprehensive utilization opportunities in the genetic field and breeding of novel varieties. Okay, next. Okay, in conclusion, recent study has demonstrated that the DNA fingerprinting of 261 varieties of cucumber with 163 perfect SNP via utilization of target SNP sequence technique So relative to conventional genotyping methods, this approach that incorporates the benefit of multiplex PCR and also high throughput screenings provide high precision, cost-effective, and also high performance. So 4 distinct subpopulations were found in 261 varieties of Chinese cucumber, which are the northern type of China, the southern type, the European type, and the Zhejiang type. So the core set of the 24 SNPs were able to discern 99% of the 261 variations of cucumber and 29 varieties of core cucumber backbone that were established in China. Therefore, the SNP sequence gold has a great potential to be used in the genetic testing and in the plant breeding process in the future. Similar techniques can be employed in the future study to evaluate genetic diversity in the different subpopulations of plants. Okay, this is our reference. And that's all from us. Thank you.